Hello everyone, I Dr. Nikita welcome you in Dr. Nikita's Pharma Online. Kindly like, share, subscribe my channel and drop your comment. So let's start next topic. So we are and what is CMC involved in the colloids. So you have three types of the colloids, that is lyophilic colloids, lyophobic colloids and the association colloids. The association colloid are or the amphiphilic colloids. These are the substances whose molecule aggregate spontaneously in given solvent to form particles of the colloidal dimension. So these are the substances when you add into liquid systems, they combine together, they form a cluster. So individually they do not impart any colloidal property, but when they associate, they cluster together, they form the clusters which is of colloidal size range, and they gives the colloidal property to the solution. So how CMC is how these clusters are forming, and what is the concentration at which this cluster is forming. Okay, so we have seen it is amphiphilic. Okay, so amphiphilic, it indicates the dual nature. So if you see this monomer means the single unit, this unit is a single unit. Okay, and this monomer is having two parts, the hydrophobic part and the hydrophilic part. This is the hydrophilic part and the hydrophobic part. So it has the hydrophilic end and hydrophobic tail. So this is water loving, and you can say this water hating. This is polar. This region gives the polar property, and this gives the non-polar property. Okay. So how the micelles are getting formed. Okay. This is the diagram where I have shown how the micelle is getting formed. But before that, I give you simple explanation for that. This micelle is similar to the human behavior. How human behaves, this micelle also behaves same. Okay. Suppose you go in a garden and you do not know whoever are there into the garden. So what you do? You do not mix with the people. You occupy one corner and you remain there in the corner till you become friendly with the other people. Okay, so when these are the many people coming together in the garden which who are single, not ready to mingle, like that. But after some time you see that these single people start talking to each other, they mingle with each other, and they become friendly with each other, and they form group. Okay, so these are the people which are forming group who were single first, but now they are together with the other people. So many people keep on coming who are single, then they mingle and they form the group, and they form many group inside the garden after becoming friendly with each other. Same things happens here in the critical micelle concentration. You see, when suppose this is the beaker you have taken, and this is water you have taken in it, and you are adding the surfactant which is acting as which is the source of monomers. So when you add surfactant, first it gets oriented at the air water interface. So this is your beaker, and where you have taken the water, here the water you have taken, this is your air interface and this is your water. Okay, so this is the interface between air and the water system. So when you add surfactant monomer in it, they directly do not go inside the water. First they occupy the boundary. Okay, so first they occupy the interface, polar head which is facing toward the water, and this is the non-polar tail which is facing the air. So when you go on adding surfactant inside it, it will keep on occupying the interface. Okay, so it will keep on occupying the interface. The polar heads are facing toward the water and non-polar facing toward the air. Once the interface is completely filled, once the interface there is no more space to occupy, then they keep on migrating inside the water. You have the water pull or water drag which is pulling this surfactant monomer inside the water. So this after complete fulfillment, they move down inside the water, and at a particular point the energy is also reduced, and they cluster together and they form the micelle. So this monomer, which was individual inside the water, they become clustered with each, they form the cluster with each other, and this cluster is called as the micelles. Okay, and the concentration at which these micelles are forming, it is called as the critical micelle concentration. And above this critical micelle concentration, if you go on adding surfactant inside the in the liquid, it forms many clusters and it changes the appearance of your solution from clear to the turbid. So you have done this experiment in critical micelle concentration determination, that when you go on adding surfactant, after some time, whatever is the clear solution you see, it becomes turbid above critical micelle concentration. So monomer, how many monomers are required to form this cluster? That is 50. So minimum 50 monomers are required to form the cluster, and the concentration at which these clusters or micelles is forming, it is called as the critical micelle concentration. This group is called as the micelle. Okay, so there are many properties which are getting affected at this critical micelle concentration point. We have seen this cluster is forming, micelles are forming. So we will see the osmotic pressure. Here is the graph where the signals are measured against the concentration addition of the surfactant. So if you add surfactant, the osmotic pressure will increase, but after CMC, the pressure remains constant. Okay. Now for the turbidity, before CMC, there is gradual increase in the turbidity, but once the micelles are formed, once so many clusters are formed inside the water, the turbidity of solution is increased. So whatever is the clear solution, you will find dense or turbid after the critical micelle concentration is reached. Now, surface tension, as you go on adding surfactant in the water, the surface tension will decrease, decrease, decrease. But particular point when the CMC is reached, the concentration remains constant and it doesn't change much. Okay. Now, for the molar conductivity, you will find that there is a decrease in the concentration, and even after CMC, there is a further decrease in the molar conductivity. Now, you have voltammetry, the parameter, you first see there is an increase, and once the CMC is reached, there is a drastic decrease for the voltammetry. Now another parameter is the solubility. You will see that when you add surfactants, the solubility increases. Solubility is increasing, increasing, increasing. But once the CMC is CMC is reached, critical micelle concentration above the solubility, there is a drastic increase or tremendous change in the solubility of the any solute. So this is for solubility, that when CMC is reached, now we will see the shape of the colloids. Okay, so we have seen that these monomers, they are forming the clusters, and they are called as the micelle. So in aqueous media, these are the spherical micelles. In non-aqueous media, these are the reverse micelle. And in above CMC, they forms the lamellar micelle. Okay. So if you take water and if you go on adding surfactant in it, so suppose this is the water you have taken. We know that water is the polar solvent, and your monomer has two parts, the polar head and the non-polar tail. So this water this beaker which is completely filled with the water, the polar heads are facing toward the water and all the non-polar tails are facing inside it. Okay, so non-polar tails are inwardly faced. So this is the spherical micelle which is formed in the water. Okay, so when you have the aqueous media or the polar media, the polar head is facing outer part, where is the solution, or the where is the polar solution or the polar liquid, and it is forming the spherical micelle. When you change this aqueous system to any other non-polar system, how the structure will be. Okay, so if this water you are changing to benzene, okay, suppose you are changing to benzene, then this becomes the non-polar solution or non-polar liquid. And we have seen this is the polar head, this is the polar head, and this is the non-polar tail. So non-polar tail now face outward because the outer system or the outer solution is now non-polar. So whatever is the polar head was outside, it becomes inside, and your non-polar tail is now facing outward. Okay, so because you have the non-polar system or non-polar solution outside, so this is called as the reverse micelle, or this spherical micelle become invert into the non-polar system and it becomes the reverse micelle. Now when you go on continue adding the surfactant above CMC, then it keep on forming the clusters, it keep on forming the micelles, and it forms the sheet of the micelles, and that is called as the lamellar micelle above CMC. And the structures look like this, like the long sheets of this micelles are forming, which is called as the lamellar sheet. So based upon your system is polar or non-polar, you will get reverse, you will get spherical shape or the reverse shape. Kindly like, share and subscribe my channel, and drop your post for any of your required topic.